In this video, we are going to do a short review of fluid and water balance. Most of our body weight is water. If you're an infant, about 75% of your body weight is made up of water. In adults, it's 50 to 60%. But as we get older, that starts to drop to about 45% of our total body weight. We have two major fluid compartments in the body. The intracellular fluid, which is the fluid inside the cell, and the extracellular fluid, which is all of the other fluid that is outside of the cell. This will include the fluid in our interstitial spaces or within the tissue. It'll include the fluid that is in our blood, so our vascular fluid or our plasma. And it will include fluids in some of our transcellular compartments, such as cerebrospinal fluid, and fluid in our body cavities like the peritoneal cavity, the pleural cavity, and the fluid in our joints. As you can see, most of the fluid in our body is intracellular fluid, around 60%. Of our extracellular fluid, most of that fluid is in our interstitial spaces, it's in our tissue. And then the next highest component is what's in our blood. So we think of the fluid in our blood, the 4 to 6 liters of blood that we have is most of our fluid, but it's really in our tissue if you look together at our interstitial fluid plus our intracellular fluid. The electrolytes in our intracellular and extracellular fluids are very different. In our extracellular fluid, so like in our blood, we have lots of sodium and lots of chloride, where the predominant ion in the inside the cell is potassium. Where outside the cell in our blood, we have very little potassium. And inside the cell, we have very little sodium and very little chloride. And so you can see in this graph, if you look at our extracellular fluid as compared to our intracellular fluid, these numbers are going to be opposite. It is important to remember that our plasma values of electrolytes are what we measure in the laboratory. So clinical values, we're always looking at what's in the plasma. That's what we can readily get to, and that's what we can measure. So when we look at potassium levels, even though most of it's inside the cell, we're looking at what's in the blood. Now, our cells, remember, create this semi-permeable membrane, and so water can move across this semi-permeable membrane. Remember that diffusion of water is called osmosis, and against a wall, like in our blood vessels, water is going to exert a force on that wall, and that's called hydrostatic pressure. It's just like what you would see in a hose that's pressing against the wall of that hose. And that can cause movement of fluid between compartments if there is a difference in concentration gradient. So as you see in this picture, there are more solutes on the right side, and so osmotic pressure is going to push water from the lower solute space to the larger solute space. And so we call this osmotic pressure. We also should understand tonicity. So this is the tension or the effect a solution that has impermeable solutes will exert on a cell size due to water movement across the membrane. This is basically saying the difference in solute concentration will drive water to go to where there are more solutes, and so that could change the size of the cell. So we're going to take a quick look at 3 different types of tonicity. So you can have an isotonic solution. That's where the concentration of water and solutes are the same as our body fluids. If you have a hypotonic solution, you have a lower solute and higher water concentration as compared to the body fluid. So you're hypotonic, there's lower tonicity. And if you're hypertonic, this solute's going to have fluid's going to have a higher solute concentration and a lower water concentration compared to the body fluid. So there's higher tonicity, hypertonic. So let's look at a picture. If you have an isotonic solution, that is not going to change any kind of water movement across the cell because the solute and water concentrations are the same. But if we have a hypotonic solution, we're going to have a solution where there are fewer solutes as compared to the inside of the cell. So water is always going to go where the party's at. It's going to go inside the cell. And so a hypotonic solution is going to cause that cell to swell. A hypertonic solution, on the other hand, there are a lot more solutes outside the cell relative to the solutes inside the cell. Again, water is going to leave the cell. It wants to go where the party's at. So it is going to go into the extracellular space now. And now your cell is going to shrink, or we call this cellular dehydration. You have lost water inside the cell. Both of these can cause problems and damage to the cell. Now let's look at forces involved in fluid exchange. So we're going to look at two main types of pressure. We're going to look at a filtration pressure, which is a hydrostatic pressure. This is the pressure that the blood exerts on the wall of the capillary. This is going to push water out of the capillary and into tissue. We're also going to talk about a capillary colloidal or osmotic pressure. This is made up of our proteins. It's going to pull water from the capillary into the tissue. There are two other types of pressures that I will mention here, but we'll focus just on our capillary pressures for simplicity. You have these same hydrostatic and colloidal osmotic pressure in that interstitial space, so in that fluid within the tissue, that is going to also have a push and pull effect. But our major pressures are going to be in our capillaries. So let's look at these, and I also think they're easier to picture. So as the blood from the arteries go into the capillary, we need to exchange and filter with our tissue. So on the arterial end, you have a larger blood pressure. So you're going to have a higher hydrostatic pressure where that blood is going to push along that capillary wall. That filtration pressure is then going to push fluid from the capillaries into that interstitial space. As you move through the capillary into the venous end, you've pushed a lot of that water out into the space. So now you have a higher concentration of proteins. And so this is our colloidal or osmotic pressure. This is made up of the solutes in our blood, primarily albumin, which is our most abundant protein. And that is going to have a pulling effect. And this is how we pull fluid from the interstitial space, from the tissue, back into the capillary, so then it can enter into the venous system for filtration. And so just to recap, this net filtration is going to occur near the arterial end because that's where this higher hydrostatic pressure is. There's a lot more water. In the middle, it's pretty even, and so you don't get net movement. But then we want to reabsorb that fluid. We want to get the waste products like carbon dioxide back into our system for filtration and waste management, and we need to get that fluid back. So there's a larger pulling pressure at the venous end because we have these proteins that have this pulling force, and so then the fluid goes back into the capillaries at the venous end. Another important thing to consider is osmolality. This is the ratio of solutes in a solution to the volume of solvent in the solution. So this is basically saying if you have some water and you pour, let's say it's 8 ounces of water, and then you pour a quarter cup of salt into that water and you stir it, you're going to have a quarter cup of salt to 8 ounces of water. So that would be your osmolality. And so we have to maintain our osmolality and our water volume within a narrow range, because remember, if we change osmolality, you're going to have water move between the extracellular fluid and the cells. And so let's look at some mechanisms that we use to regulate osmolality and water intake. So our main hormone we're going to talk about here is antidiuretic hormone. It's also called vasopressin. It controls water balance and osmolarity. It's produced by the hypothalamus, and it is released by our posterior pituitary. Its major actions are constriction of our arterioles, which increase blood pressure, and it increases reabsorption of water in the kidneys. So this is how this works. If your blood has a high osmolarity, so there's a lot of solutes, maybe you're dehydrated, that is going to stimulate your thirst mechanism, and that's going to drive you to drink water. That will then increase your extracellular water volume and decrease that serum osmolality. At the same time, that is going to signal the release of your antidiuretic hormone from that posterior pituitary. ADH is going to travel to the kidney, where the kidneys are going to hang on to more water. And again, that's going to dilute that blood and decrease that osmolarity. If your osmolarity is low, maybe you don't have enough salt in the body, you're not going to be thirsty, and so you're not going to drink as much water. You see this a lot in older adults. And your ADH is going to cut off, so you're not going to hold on to water. And so you're going to try to concentrate the blood by excreting water out through the kidneys into your urine so that you can then increase the concentration of that blood. So that's just a quick overview of water balance.